Hello, everyone. My name is Professor Pauline Mackey. I would like to welcome you to the International Menopause Society webinar on the microbiome and nutrition in midlife women. We have two fantastic speakers today, Dr. Kelsey Gabel, who will give a lecture on intermittent fasting for the postmenopausal women, and Professor Jack Gilbert, who will discuss menopause in the gut microbiome. I'd like to first thank you all for coming here today, and I'd like to thank our sponsor, Besson's Healthcare. This webinar was supported by an unrestricted educational grant from them. They had no role, however, in the selection of topics, the selection of speakers, and have not vetted or reviewed the content of the speakers' presentations today. So today we will begin with a presentation by Dr. Kelsey Gabel. Dr. Gabel is an assistant professor in kinesiology and nutrition at the University of Illinois at Chicago. She received her PhD in human nutrition from that institution, and she's been studying intermittent fasting for almost a decade and has emerged as a leader in that field. Her research is focused on how intermittent fasting may mitigate weight gain and adiposity, adverse effects and metabolic dysregulation during anti-tumor treatment for breast cancer. She looks to develop transdisciplinary work exploring how alternatives to daily calorie restriction can be leveraged to improve the long-term health and quality of life of women as they age. Following Dr. Gabel's presentation, we will have one by Professor Jack Gilbert. Professor Gilbert earned his PhD in 2002. He's currently at the University of California, San Diego, where he's professor of pediatrics and the Scripps Institution of Oceanography, associate vice chancellor for marine science, and director of the Microbiome and Metagenomics Center. He co-founded the Earth Microbiome Project and the American Gut Project. In 2017, he co-authored Dirt is Good, a popular science guide to the microbiome in children's health. In 2018, he founded BiomeSense, Inc. to produce automated microbiome sensors. And in 2023, he became president of Applied Microbiology International, the oldest microbiology society in the United Kingdom. And I welcome both of them to our webinar today, and I will turn it over to Dr. Gabel.
Good morning or afternoon, wherever you are. Thank you for having me today. I will be discussing intermittent fasting for the postmenopausal woman. So today we will be discussing intermittent fasting, the background of intermittent fasting, alternate day fasting, time-restricted eating, and then hopefully our future directions. So the first thing I want to discuss is what is intermittent fasting. This has become more and more popular, but it really is an umbrella term. So I wanted to get a few of the semantics out of the way. So this really encompasses 3 diets, and the first is called alternate day fasting, and this is where you eat between 0 to 25% of energy needs on one day, and then the next day people are able to eat whatever they want, and they repeat that pattern throughout the week so that overall there is a calorie deficit throughout the week. The next is the 5-2 diet, and this is very similar to alternate day fasting. However, fasting happens only 2 days out of the week. And then the last, this is probably the most popular currently, is time-restricted eating. And this is, again, where we shorten the daily eating window in which people consume calories and extend that overnight fasting period, again, producing an overall calorie deficit. So I wanted to start with alternate day fasting because this is really where the intermittent fasting research started 20 years ago. And we did recently release a study. So we wanted to look at alternate day fasting with exercise or exercise alone or alternate day fasting alone to treat non-alcoholic fatty liver disease, which is tightly correlated with insulin resistance. And so we did have 20 participants per group. And remember, one day they're fasting, the next day they are eating whatever they want. They were prescribed 5 days a week of supervised exercise. And so our participants were middle-aged, mainly female. And looking at the adherence data, adherence to alternate day fasting was relatively high. So the adherence here you can see was about, or this is the exercise. They exercised about 4 out of the 5 days a week that was prescribed, which I believe is pretty great. These individuals were sedentary prior to the start of this study. And then here you see the fasting day energy intake. And we found our participants, they were prescribed 600 calories based on their energy needs, and they ate about 673. So they were within that range, and we did see weight loss, so we do know that they were calorie restricted throughout the trial. We did look at liver fat through MRI, and we saw that the intrahepatic triglyceride content decreased significantly in the combination group versus the exercise group and the control group. However, it wasn't significant versus the alternate day fasting group. We also wanted to look at body weight, since this is correlated with non-alcoholic fatty liver disease as well as insulin resistance. And again, we saw that combination group lost significantly more weight than the exercise and control, but not versus the alternate day fasting group. And we were a little surprised by these results, that both groups kind of lost the same amount of weight. However, I think that the exercise group probably had an increase in hunger, and so they might have compensated a little bit more on those feeding days versus the diet alone. So while the weight loss isn't more significant, there are obviously other benefits to the exercise other than the weight loss alone. And then we did look at liver enzymes, ALT and AST, and we saw a significant decrease in the ALT for the combination group, but only versus control, because here you say the alternate day fasting also improved their ALT as well. Looking at these metabolic disease risk factors, which we thought would decrease with the non-alcoholic fatty liver, we also saw insulin, fasting insulin, waist circumference, and insulin sensitivity and insulin resistance improve in the combination group, again, versus the controls only. And that's again because you see these other improvements in the other groups with the exercise and the alternate day fasting. So it's only significant for the combination group versus the controls. And this, I think, is what is really interesting. So we did run the largest and longest alternate day fasting trial during my master's work. And we saw that 20% of our participants gained weight doing alternate day fasting. About 40% lost 1 to 5%. And 40% lost 5 to 15%. We cut at that 5% because that seems to be clinically significant, where we see reductions in lipids and insulin resistance. And the interesting thing is we compared this with calorie restriction, and we saw the exact same pattern. So this is our daily calorie counting, where 20% gained weight doing calorie restriction, about 40% lost 1 to 5%, and then 40% lost 5 to 18%. And why I think this is really important is it's important to look at these alternative diets, because one diet is not going to fit all people. So we were also interested in who was most successful doing alternate day fasting. And we didn't see any differences between males and females. But here, I think, is the cool part. The most successful participants were aged between 50 and 59, which is a time where I think most people struggle with weight loss. So I thought this was really interesting that that age group was the one that really had the most success. So I want to move on to time-restricted eating just because it is kind of the hot topic of diets right now. So I'll take you through what we're finding and where I hope we are going. So again, remember, there's no calorie counting in time-restricted eating. You're just watching the clock. You don't have to measure or weigh food or track it. There is no diet quality restrictions. So while we might not be improving the quality of the diet, it is very accessible. So people who may not have the means or the access to changing these diet quality patterns could still improve their health. So this was the first study that we did in people with overweight or obesity. And we didn't, because it was the first study, we didn't know where to place the eating window. So we kind of just put it smack dab in the middle of the day. Now I think the most popular window is noon to 8, but this is just where we started. So we did see that time-restricted eating was easy to adhere to. So people were adherent about 6 out of the 7 days of the week, and they adhered through that. They maintained that through the entire 12 weeks of our trial. They did lose a significant amount of weight versus the control. So they lost over 2.5% of their body weight in 12 weeks. We do see this kind of stark decrease in the first month and then slow and steady weight loss thereafter, which I think people would be sort of discouraged by this initially, but 1 pound a week is kind of what I look for as a dietitian anyway. And can be more maintainable. And remember, these individuals did not track or manage their food. They literally just followed the clock. So they were able to eat whatever they want, and they still had this weight loss result. We also wanted to look at diet quality, which did not change. However, they did eat less. These were based off of food records. So we know there's issues with food records, but hoping the ratio of underreporting is the same between baseline and week 12. And so they ate about 350 or 20% less. And that has been seen in multiple time-restricted eating studies since. So with 8 hours, individuals eat about 20% less. With cardiometabolic risk factors, systolic blood pressure did decrease significantly versus the controls. And that has actually been seen since, independent of weight loss. And then we weren't powered to see changes in insulin. However, there does seem to be something there, and it's kind of goes back and forth in trials since this one. So overall, we did see a significant weight loss, a high sustained adherence, unintentional calorie restriction of about 20. And we did look at eating disorder symptoms and adverse events and did not see any changes there. Systolic blood pressure decreased. There were no change in plasma lipids, and glucoregulatory benefit was kind of unknown. So then we thought, what if we shorten the window to 4 or 6 hours? Would that be doable? Would it be more beneficial? So we started to shift the window more towards that popular time because a lot of our participants told us they wanted to eat dinner with their families. That was most important. And because Chicago, you commute, they weren't able to get home by 6 o'clock. So I think this is really interesting that more is not always better. So the 4 and 6 hour window both lost the same amount of weight in the 8 weeks. So they both lost 3% of body weight. This weight loss seemed to be a little bit more aggressive than the 8 hours, but not a ton. So people can still do the 8 hours. And if they want to kind of get a little bit more bang for their buck, they can go down to the 6 hours, but I wouldn't really recommend the 4 hours. They again were adherent 6 out of 7 days of the week. And fasting insulin and insulin resistance here did improve. This was interesting because we have seen systolic blood pressure decrease in other trials independent of weight loss. And here the blood pressure, heart rate, and lipids did not change. And they saw a little bit more of a calorie restriction. So while 8 hours was about 20%, 4 and 6 hours was about 30%. So we broke this down in pre and postmenopausal women, and we saw that they really lost the same amount of weight. The postmenopausal group had a higher visceral adiposity, which we would expect to see, but they also lost more visceral adiposity during the trial. So it really brought them to the same level of visceral adiposity as the premenopausal group. We also wanted to look at these metabolic risk factors to see if there was any difference pre and postmenopausal. And while we saw changes in insulin and HOMA-IR, those were the same over time between pre and postmenopausal women. And then there were no other changes between groups over time. We also wanted to look at sex hormone changes in premenopausal and postmenopausal women during this trial, and we didn't see any changes other than in DHEA, which does decrease with age. And so DHEA has actually been used as a supplement to treat postmenopausal menopausal symptoms. So this may actually be beneficial in some way during or after menopause to kind of decrease some of those symptoms. So overall, 4 and 6 hour time restricted eating, they lost about 3% body weight in 2 months versus the 3% body weight in 3 months with 8 hours. There was no difference between menopausal status and weight loss or any of the other cardiometabolic parameters. It did have a high self-reported adherence and about a 30% or 500 calorie per day caloric deficit. Insulin and insulin resistance did improve. Oxidative stress, I didn't show you those slides, also improved, but there were no impact on lipids or inflammation. So to touch on a few of our future directions, we are running a trial right now with time-restricted eating during chemotherapy for breast cancer, which we are adding a Mediterranean diet arm as well to see if we can improve that diet quality using a Mediterranean style diet versus standard care. And we'll look at those cancer biomarkers as well as cardiometabolic blood markers. We have 7 that have gone through the trial. 1 is still, and then we're continuing to reach our sample size. This study will be running hopefully soon of resistance training and time-restricted eating with resistance training. This will only be in ages 60 to 85. And we wanted to look at aging is such a privilege, and we really want to look at how we can maintain the function of life as we age. So I was thinking how great it would be to add resistance training to look at metabolic markers and pre-diabetes as well as cognition going forward. And we'll also look at sex differences in this group to see any of those changes. And then last, which is a great way to stop for Dr. Gilbert, we are adding a prebiotic fiber supplement to see if we can harness the gut microbiome during time-restricted eating in pediatric oncology survivors to see if short-chain fatty acid synthesis can improve some of those hunger hormone control as well. So we're doing time-restricted eating with a fiber supplement, and then ad libitum in pediatric cancer survivors. So I'd like to thank my mentors as well as these funding sources and collaborators. And I am looking forward to our question and answer soon, but I want to thank you. And if you do need anything, please feel free to reach out via my contact information. Thank you so much.
Thank you. Thank you, Dr. Gabel. And please put questions for Dr. Gabel in the Q&A, and we will entertain those questions after Professor Gilbert. And now, Professor Gilbert, we'll turn it over to you.
Fantastic. Thank you. That was an amazing talk. I think you and I have a lot to talk about, Kelsey. A lot of the work that we do is focused on trying to understand how diet manipulates and alters human health. And so it's fascinating for me to be able to explore opportunities that will allow us to augment diet changes with particular strategies, right? So I can imagine combining an improvement in diet, such as your fiber supplement, with time-restricted eating in order to improve other characteristics. So we should talk. So my name is Jack. I'm actually a professor of pediatrics and oceanography, which is kind of strange. But at UC San Diego, we tend to combine talents and skills across departments and institutions in order to create new opportunity. And so a lot of the work that we do is focused on trying to understand how the microbial communities inside our bodies and outside our bodies influence health. And as we live in a coastal town, as you can see from the view behind me, this is actually one of the views from my office. Understanding how the environment around shapes our health is vitally important to us. So I'd like to start just by talking about how the microbiome and the immune system function. This might be strange when you're talking about the endocrine system, but trust me, everything is connected. And I think this is the primary interface. So the immune system is historically been seen as a little bit of like an army, right? It's an army that goes in, wipes out the bad guys. But it's much more like a national park service or a gardener, right? It's there to manage that microbial garden inside your body and to manage the microbial exposure that would come and disturb your garden, right? You're being bombarded with about 30 million different microorganisms every hour. You are also emitting into your environment about 38 million microbes every hour. So there's a net growth deficit in terms of immigration. And that immune system has evolved to manage that exposure. In fact, the immune system in your skin is sampling the microbes which you are exposed to and which grow on your skin every second of your life. So that should any of those microbes on your skin get inside you, the immune system can rapidly take care of them with a learnt immune response. But it's also there inside the gut, inside the urogenital tract, inside your lungs, inside your oral cavity, on your skin, in order to try and manage the microbial organisms which it's become dependent on. So every single one of these bugs, as Kelsey pointed out, are producing chemicals which can be vital to our health. So they break down your fiber and they produce short-chain fatty acids like acetate, propanoate, or butyrate, which can dampen down immune responses or change leptin hormone production, as Kelsey was pointing out, and reduce hunger. These characteristics are really important, and the immune system is there to manage that microbial garden and make sure it produces the right kinds of chemicals. So what happens when that goes wrong? Well, let's take two states here. And this is a wonderful infographic from Mariana Bindlos, who presented this just 5 years ago. But I think it's a fantastic opportunity for an educational moment. So let me walk you through this because it can be a little bit complicated. But this is a cross-section of the gut. This is the lumen of the gut up here. And the blue color represents a low oxygen potential. So not much oxygen, it's anaerobic, and that promotes the growth of obligate anaerobic bacteria, such as the ones that ferment your indigestible fiber and produce those short-chain fatty acids. In this case, the short-chain fatty acid I'm most interested in is butyrate. Butyrate is a fuel for beta oxidation pathways in the colonocytes, those cells that line your intestine. And when beta oxidation goes on, these colonocytes become very mature. They form tight junctions between each other and stop bad things from the gut getting into the body. You've heard of leaky gut. Well, that would be our situation on the right. So on the right, we have dysbiosis. This is what happens if you eat an unhealthy diet, such as a high saturated fat or high sugar diet for a prolonged period of time, or you temporarily have to take an antibiotic, a non-absorbable antibiotic like vancomycin that can destroy certain organisms and keep the antibiotic resistant one alive. Many of those antibiotic resistant organisms, or the ones that love simple sugars, are facultatively anaerobic bacteria that produce things like lipopolysaccharide, LPS, hydrogen sulfide, and flagellin. These are proteins and chemicals which can stimulate inflammation in the gut, but they also don't produce a lot of those short-chain fatty acids from the digestion of fiber. And so the colonocytes have to rely on anaerobic glycolysis, and they become relatively immature, and the junctions between them start to degrade. So stuff can get inside, like that LPS and flagellin, and cause inflammation. But also these immature colonocytes don't absorb oxygen from the bloodstream. This now leaks into the gut cavity, raising the oxygen potential and preventing our obligate anaerobic friendly bacteria from growing back. That's dysbiosis. These are two cycles. On the left, we have a positive feedback loop. The body and the microbiome are working as one, as they were supposed to. On the right, we have dysbiosis, where a disruption in the microbiome is causing a disruption in our immune system and a disruption in total physiological response. So dysbiosis, we think, could influence endocrine disruption, especially in perimenopausal, postmenopausal women. So bile acids secreted in the gut secrete conjugated estrogen and phytoestrogens that are deconjugated by bacterial beta-glucuronidase. So here, for example, I have bacterial beta-glucuronidase activity, which is taking that conjugated estrogen and phytoestrogen and deconjugating it into chemicals, which can then be reabsorbed from the gut into the bloodstream and have receptors at very distal areas, such as the vaginal epithelium. If I have a disruption in this beta-glucuronidase activity, I'm going to have a reduction in the production of these deconjugated estrogens and phytoestrogens, which will then disrupt these distal processes. For example, in this context, causing a reduction in certain contents, like the epithelial thickness will go down, the mucus secretion in the vagina will go down. We'll get an increase in pH and a decrease in glycogen, which are symptoms common in menopause and postmenopause. So obviously, I've talked about the reverse of these arrows, because the reverse of these arrows is what happens when these chemicals are no longer being produced by bacteria in the gut. So dysbiosis may reduce estrogen metabolizing bacteria, causing a decrease in these circulating estrogens, which then influence hypoestrogen related diseases, such as a reduction in vaginal epithelial thickness and mucus secretion. This paradigm is fascinating because it does provide us with an opportunity to understand what happens in the gut and how it affects the rest of the body. So dysbiosis can influence that endocrine disruption. There's a fascinating role also during postmenopause of the vaginal microbiota and its association between that and the symptoms of menopause and postmenopause. So there's an inverse correlation between the lactobacillus organisms, which produce that low pH in the vagina and are incredibly representative of the vaginal microbial community in healthy individuals. So when you have an inverse correlation between lactobacillus and dryness and an increase in bacterial diversity in women experiencing moderate to severe vaginal dryness, you should pay attention, because there's something going on inside the vaginal community which is no longer in homeostasis, which is now disrupted to dysbiosis. So this composition is actually way more stable than premenopausal populations, but also highly diverse. So we're getting a lot more organisms growing, and a lot of these lactic acid producing bacteria, lactobacillus, which are driving down the vaginal pH, have disappeared. And so this could be either a cause or a correlation. If we just go back to this one image, if I have a disruption in the gut that produces chemicals that get into the bloodstream, which influences the vaginal epithelia, reducing the mucus secretion, which is a lovely foodstuff for those lactobacillus, then I'm going to get a reduction in the lactobacillus and an extreme growth of other organisms, which will take advantage of that system. And that's what you see here on the right. These these participants not only have dryness and a significant increase in microbial diversity, they also have a significant increase in vaginal epithelial inflammation. Why? Because a lot of these lovely, beautiful, colorful bugs, not the nice blue ones, which are our lactobacillus, but these beautiful, colorful bugs are also those ones that are producing lipopolysaccharide, hydrogen sulfide, and flagellin, such as from our gut story. These are facultatively anaerobic organisms which love living in mucus-rich environments, but also can survive in pretty much anything. And they produce a lot of pro-inflammatory compounds. And in fact, we're working, one of my graduate students now is working with trans men who obviously, due to the use of testosterone or T, have significant problems with vaginal tissue atrophy and dryness, and therefore are experiencing a almost rapid onset menopause situation for their particular condition. And they present with nearly exactly the same paradigm: significant reduction in microbial beta-glucuronidase activity and a significant increase in vaginal microbial diversity, which associates with dryness and atrophy. So microbial changes also shift with endocrine activity. So menopause in the gut is associated with a reduction in microbial diversity. So it might be associated with an increase in microbial diversity in the vagina, but inside the intestine, we're actually seeing a reduction in microbial diversity and an increase in the pathogenic E. coli and beta-glucuronidase. So we see postmenopausal microbiome is more like the male microbiome than the female premenopausal microbiome, which is absolutely fascinating. So a turning off of these hormone states inside the body can shift the gut microbiome into something which is much more androgynous, if you will, and much less like we would normally experience in premenopause. And these menopause-related gut species functions, such as beta-glucuronidase and sulfur metabolism, tend to influence these serum-level progesterone steroids post-menopause. So our premenopausal gut microbiome is more diverse, less similar to men, and has higher estrobiome potential. It can do a lot of that beta-glucuronidase active metabolic capabilities. And then our postmenopausal microbiome is less diverse, more similar to men, and has lower estrobiome potential. So this can influence cardiometabolic health via a number of different pathways, which I won't go into now, but if you're interested, I'd be happy to answer questions later. Metagenomic analysis does demonstrate a significant shift in the gut microbiome with menopause. In the gut, I mentioned the vaginal community before, but this is in the gut. So menopause tends to be associated with a depletion of Clostridium lactiformans, which is associated with a higher HDL cholesterol and lower waist circumference. So for example, if you have in premenopause a lot of Clostridium lactifermins and you have a higher level of HDL cholesterol, which is good, if you remember, and a lower waist circumference, which is generally considered good, then post-menopause, you're likely to see a reduction in that organism and an increase in those factors. You can see that here. That's the Clostridium lactifermentans there, because it ferments lactate. And we see a significant positive correlation with HDL cholesterol and a significant positive correlation with HDL cholesterol over here. And we also see a significant negative correlation with waist circumference here. And then after menopause, we see an enrichment for Sotorella wadsworthiosis. That's this organism up here, which has a significant correlation with a higher diastolic and systolic blood pressure, and in both of these populations. So, well, this is interesting, right? And if the microbiome in the gut can be disrupted, what other functions could it absolutely influence? Well, we also explore how the microbiome changes our mental state, our health, in terms of depression and anxiety, both throughout life in the pediatric space and in perimenopausal, postmenopausal women, and through age. And we know through many fantastic research studies that the microbiome is linked to things like brain development. It's correlated with brain development. It's correlated with mood and behavior, and it's correlated with those neurodegenerative disorders such as Alzheimer's and Parkinson's that you'll get later in life. And there's also a comorbidity quite often between these central nervous system disorders and GI disorders. So, if you've got diarrhea or constipation, you're often going to have things like anxiety and depression. And then those antibiotics use can be linked to behavioral phenotypes such as depression and anxiety. So knocking out bacteria can have an impact upon depression and anxiety. But interestingly, antivirals and antifungals have no such impact on depression. And we also know over here in this lovely infographic with red and green mice, I apologize for the color choices for those who are colorblind, but in the top we have clinically depressed people. If we take clinically depressed microbiota and put it into an animal, the animal will exhibit depression-like behaviors. And we can argue about those, but I'll show some in a minute. And non-depressed microbiome, when it's transferred into an animal, will show no such change in behavior. And interestingly, there are multiple pathways by which this disruption in what goes on inside the gut can influence what goes on inside the brain, such as the modulation of neurotransmitters, inflammation pathways, inflammation being key. Remember, our immune system is our gardener, and if the microbiome is disrupted, it's going to disrupt the immune system throughout the body. And direct signaling by this vagus nerve or efferent spinal pathways, which can lead to brain stimulation and changes in neurotransmitter activity up there. We did a study in an animal-to-animal model examining whether fecal microbiome transplants are taking the poop from a healthy animal and putting it into an unhealthy animal, quote unquote, would influence behavior. So we took, in this case, rats, which have a wonderfully similar pH in their gut system to humans. So we tend to see representation of gut activity very similar to humans. We took Sprague-Dawley rats, SD rats, which are considered non-depressed, and then we took fawn-hooded rats, which are considered depressed, right? I'm using quotation marks because obviously we're not inferring a human neurological phenotype on these animals. We are trying to use their behavior as a model to see if we can change the microbiome to change behavior. However, pre-FMT, we can look at these 3 tests over here. The forced swim test, where we put a rat in a jar of water, and if it swims for its life, we consider it happy. And if it just gives up and lays on its back in the water, like looking like a bored fish, then we consider it unhappy. And indeed, our fawn-hooded rats spend more time floating than our SD rats. And if we take the microbiome from a fawn-hooded rat and put it into another fawn-hooded rat, that doubling down shows a similar kind of depression phenotype to our original fawn-hooded rats. But if we take the microbiome from an SD rat and we put it into a fawn-hooded rat, we see a recapitulation to a certain extent, significantly, of the SD phenotype in our fawn-hooded animal. So we can change its behavior. Similar thing with open field, which is testing for anxiety. Our SD rats tend to be more active in the open field and less anxious. When we take a fawn-hooded rat and give it an SD rat's microbiome, it becomes the FH rat becomes less anxious. And in the sucrose preference test, we get a doubling of this effect. So fawn-hooded rats that get an SD microbiome tend to much more prefer sugar, right? So we can induce this sugar craving, this glucose craving, or sucrose in this case, craving in these animals by giving them a SD microbiome. Interestingly, serotonin in the serum isn't really affected, despite the fact that we know that the SD microbiome produces a lot of serotonin in the gut, which gets into serum, but we can't recapitulate that in fawn-hooded rats that get an SD microbiome. However, serotonin and norepinephrine and other neurotransmitters in the hippocampus can actually be influenced. So fawn-hooded rats that get an SD microbiome show an increase in serotonin and an increase in norepinephrine in the hippocampus. So there's an association there between changing the microbiome and altering both behavior and neurotransmitter production. So what do we think is happening? When in our fawn-hooded wild-type rat, we have a gut microbiota that's producing a lot of carbon-rich metabolites. These carbon-rich metabolites are absorbed by the gut lining into the bloodstream, and these carbon-rich metabolites appear to transgress the blood-brain barrier and actually influence neurotransmitter inflammation and various other functions in the brain. Okay, great. These carbon-rich metabolites appear to be produced by a bacterial group called Dialysta. And when we take a fawn-hooded rat and we give it an SD rat's microbiome, we see a significant reduction in diet ester among other organisms. This reduction in these organisms is associated with a significant decrease in enzymes associated with carbon metabolism, a significant decrease, again, in carbon-rich metabolites in the blood. So yeah, we've knocked out these bugs which produce these carbon-rich metabolites, a significant increase in nitrogen-rich metabolites, which then cross into the blood-brain barrier, into the brain, and have an impact upon both a reduction in hippocampal osmolytes, a reduction in neuroinflammation, the brain is less inflamed, and a significant increase in those neurotransmitters, as we saw previously, serotonin, norepinephrine, but also dopamine. So these relationships appear to be mutable. And if we can mutate them by changing the microbiome, we can also alter brain activity, and that could affect human behavior. I won't talk about that work at this time. So just to finalize, we've been looking at the microbiome for 300-400 years without really realizing it by looking at the state of the gastrointestinal contents that leave our body. So this is a Bristol stool chart, which shows everything from diarrhea to constipation. And we look at it in a bowl. All we're really doing is taking this information and molecularizing it so we can understand it more effectively by looking at the bacteria in the poop. We do this through microbiome-wide association studies, which is akin to a genome-wide association study, where we would look at a genetic allele, a variant in a population, and see if it associates with disease. We do exactly the same thing, just with microbes and microbial metabolites. So they produce this cornucopia of metabolism inside our body, which is vitally important for our health. This cross-sectional analysis helps us to really pinpoint those variants. But we just found out through work from one of my ex-graduate students that the fourth dimension, time, is vitally important. And in fact, this graph shows the probability that in a population you will identify a particular biomarker associated with a particular disease in the gut based on the number of times that you collect their poop. So if I collect their poop just once, I have a huge amount of variability in my ability to predict a biomarker. And to reduce that, I really need 15 to 20 consecutive samples to reliably get statistical power to predict a particular biomarker in disease. Why is this so important? Well, unlike things like what height, weight, and age, and blood pressure and insulin, like we heard about in the previous talk and earlier in this talk, which are very low dimensional and can be static, numbers don't change very much, or highly dynamic, they change hour to hour. Or like genetics, our own genome, which is pretty static, doesn't change much throughout our life, but is very high dimensional, tons of information in there, right? The microbiome is like your worst case scenario. It's highly dimensional, there's tons of information, about 100 times more genes than the human genome, but it's also very dynamic. It changes when you eat or when you restrict your diet, as we heard from Kelsey. So to effectively discover biomarkers, we need a huge cohort size and frequent sampling, which makes it incredibly complicated. To solve this problem, as we mentioned, I formed a company, which I do have a small financial interest. And in this company, we're producing an automated microbiome lab built into a box, which sits next to your toilet. And then in our clinical trials, our participants put in a number in the top, opens up a little flap. They drop a piece of used toilet tissue in there. No mess, no fuss. They don't even have to look at their own poop. They can just fold it, drop it. And it extracts all the DNA, RNA, and metabolites from there and stores it in a single barcoded sample jar. Or sample well. And it can do this for up to 60 consecutive samples, and it can preserve them for up to 3 or 4 months in a highly reproducible way that removes all of the mess. People don't have to look at their poop. There's a poop in a hat and scoop it out, and all of the other issues associated with microbiome sampling, such as shipping and variation in how a sample is extracted in a lab. And we're applying this into our new NIH $170 million precision nutrition study, of which we are the Microbiome Metagenomics Center. And so in here, we will be analyzing the microbiome and nutrition in 10,000 people and putting about 3,000 or 4,000 of those through very rigorous crossover design dietary interventions to understand how individual people respond to their diets in unique ways. And in this, we are applying quantitative for the first time metagenomics and metatranscriptomics. Usually metagenomics and metatranscriptomics are not quantitative, they are quite proportional. We can't really tell how many bugs you have, just what percentage of our data showed that bug. Well, we've changed that paradigm with new innovations. So what can the microbiome do for us? Well, it can help us to develop these diagnostics to predict disease and complications before they happen. We can commercialize IP around small molecules for the treatment of lots of disease. We can educate antibiotic stewardship and best clinical practices by looking at what microbes in your gut are affected by antibiotics. We can redefine that environmental health nexus, how our environment is shaping our health, which can lead to shifts in translational research. It can broaden the clinical trials to test microbiome interventions in high-risk diseases. And it creates new technologies and informatics to support the globalization of diagnostic and therapeutic potential. Thank you very much for listening. It's my wonderful lab down here at Scripps Institution of Oceanography. And I'll take any questions.
Lovely, lovely presentations. Thank you. Thank you, Professor Gilbert. And thank you again, Dr. Gabel. We have a lot of questions from our audience. And without further ado, why don't we begin? Okay. So just a clarification from one audience member, Dr. Gabel, were the participants in the TRE study exercisers or sedentary?
They were sedentary.
Okay.
Yep, prior to the start.
I'll follow up with actually the question that I asked you the first time I listened to you speak. Can I have coffee during my fasting period? Can I have water?
Yes. Oh my gosh. Yes. I have tried all of these and I don't know that I would survive these fasting diets without coffee. So we do allow anything that is calorie free during the fasting window. We do try to limit diet sodas to a maximum of 2 a day. But coffee, tea, as long as they don't have sugar and milk added, are good to go. So I have not eaten yet, but I have had my 2 cups of coffee.
Yeah.
We also know diet sodas can disrupt hormonal activity and disrupt insulin capabilities.
Fascinating. Thank you. So Professor Gilbert, how can one become a participant in the All of Us study for continuous stool sampling?
You can actually sign up. We have it's only in America, unfortunately, at this time, but there are 6 sites around the country. So Chicagoland. So for both of you, I hope you will consider signing up. And for any of our audience members, we have populations being recruited in the Northeast, Southeast, down in Florida, Georgia, and North Carolina, Boston area, Chicago, in Texas, in LA, so Southern California, Northern California, Oregon, and also in Washington State.
Fascinating. It's fun to project forward and think about precision medicine in the future when we look at those innovations. Congratulations.
Well, also, I would love to see if the reason, Kelsey, you're getting these variations in how people respond to these diet trials is also associated with what they're eating at the time, right? And so, you can, again, precision diet combined with these types of restricted approaches could have a huge impact.
Absolutely.
Yes. Dr. Gabel, can diabetics safely do intermittent fasting?
Yes, is the short answer. There have been a couple of studies using 5-2, and we are actually at UIC wrapping up a time-restricted eating trial with people who have diabetes. We do have to decrease their short-acting insulin on the fasting days for alternate day fasting, and then decrease those by a percentage during the time-restricted eating. But we're actually finding that they are doing, so we're running, it's people with diabetes with time-restricted eating versus calorie restriction, and they are much more successful using time-restricted eating because most of us have tried calorie restriction for so long that we're kind of burnt out. Yeah. And this is something new. And so they do seem to be more successful, but I think there's more to come. Definitely under the advice of a doctor, I would see a dietitian and a doctor while doing any of these fasting with, especially if on insulin.
Thank you. Professor Gilbert, how significantly does gut dysbiosis contribute to the total estrogen pool in postmenopause? And is that magnitude of effect, that hypoestrogenism, does that contribute to symptoms?
It's a really good question, and one that we're only just starting to be able to answer with larger cohorts. I would say that our existing cohorts that have been either published or have been analyzed and not yet published are low powered to actually be able to characterize that particular contribution. I quantify the contribution effectively because it does have a quite high variance margin between people. But if I was to spitball it, I'd say that it can contribute to between 50 and 20% of total estrogen inside the body.
Fascinating. Okay, Dr. Gabel, we have an attendee here whose reflexive habit is one of time-restricted eating, and she's wondering, because she's transitioning through the menopause, she's still noticed that her waistline is she's gaining weight nonetheless, even though this has been her kind of default lifestyle. What else would you recommend that she do in that case?
Well, I would definitely recommend talking to a dietitian to look at diet quality changes. Exercise, I saw there was exercise in there. That would be excellent if it goes along with your lifestyle, anything that you enjoy. It doesn't have to be this kind of structured right or wrong exercise, just move in a way that brings you joy. And then I don't know if Professor Gilbert has anything to say, but maybe there is something that the microbiome could also be harnessed here. And then I would say bodies will change, and this will not be the end all be all. It will probably change again. So yeah, you could definitely look at diet quality. You could shorten the window for a while, or maybe try alternate day fasting.
Thank you. A question for Professor Gilbert about skin dysbiosis. Can we talk a little bit about that? And what is the effect on transdermal estradiol? I'm wondering here if they mean the absorption, or if perhaps even they mean the effect clinically of transdermal estradiol in that condition.
Yeah, actually, we are exploring that in a trial right now to try and understand if variation in the microbial signature on the skin influences both absorption and also any associated clinical side effects or clinical manifestations of changes in phenotype. We are not right there to be able to recommend or say any particular conclusions. However, there does look to be an association, but I don't have any statistical data to present at this time. But yeah, it's whoever's asked that question, that's a really good question and one we're very, very interested in.
Thank you. Dr. Gabel, are there benefits to intermittent fasting for those who are not overweight?
That's a great question. I don't think there is enough data yet to know for sure. If you do have a higher blood pressure, I think it could be beneficial. We're not sure if it's just that individuals are drinking more water because of the extra fasting time. However, right now, it looks like the benefit of time-restricted eating is the calorie restriction itself. We did actually look at microbiome in that first study, and we weren't powered to see those changes. So we didn't actually see any changes. I think that will be the key to this, to be honest, is to see if those other factors of that feed and fast pattern actually have some other benefit. Some people report that they have better sleep. Some people report that they, have less constipation. So we'll see. I don't know. Again, I don't think we're there yet, which is, to me, really exciting to be able to look into those questions, but I just don't think we're there yet to know.
Thank you. Professor Gilbert, do you think it will be possible eventually to alleviate menopausal symptoms using probiotics that enhance the estrobolome?
It's a cycle, right? So obviously the body has to be producing enough conjugated bile acids that contain the estrogen or the phytoestrogen and that estrobolome component in order to drive microbial or for microbial beta glucuronidase activity to actually have an effect. So it's hard to know whether just increasing microbial beta glucuronidase activity would actually have a significant impact in the changing body, right? However, we are interested in testing that hypothesis and also exploring how it could influence symptoms associated with transition for trans men as well. And so there is an ongoing study to explore whether different probiotics with improved beta glucuronidase could have an impact upon some of those symptoms. Again, this is such an early scientific area, very few studies. We're really excited by those findings.
Thank you. Dr. Gabel, what about intermittent fasting in children? We have this big obesity problem in the US, so much so that they're actually recommending now that children start medications for obesity. Can you comment about intermittent fasting as a means of addressing this pandemic?
Yeah, so as of right now, we don't recommend intermittent fasting for individuals under the age of 12. And if 12, they need to be in that 98th percentile of BMI or height and weight. I would still, as a dietitian, all of this makes me a little nervous because this is the time where kind of those disordered eating patterns in preteens and teens kind of arise. However, I do think there are some interesting concepts as a family that you could do. So maybe there's no eating after dinner. So dinner is the last meal of the day, the family dinner, and then there's no snacking at night. Things like that, I think, could be utilized from time-restricted eating. However, there has not been any. I think there was one study in individuals that were 12 years and older in Australia, but that's the only study that's ever been run. So we're just not sure about the safety, and I personally wouldn't recommend it.
Thank you. Professor Gilbert, big question for you. Can probiotics improve the microbiome?
Most of the probiotics that we currently have available today, their main activity is through stimulating the immune system. And so there is some compelling evidence to suggest that just stimulating the immune system as the gardener of the microbial body can play a role in improving the microbiome, but it's limited, right? And generally the probiotics that you consume are pooped out the next day. And so don't take up residence in the gut environment unless there's some other factor involved. So I would argue the jury's out. It's dependent upon the people, and there's a huge amount of variation in their potential impact.
And at a follow-up question, at the, if you will, prevention side of things, is it critical that we eat things like Greek yogurt or other kinds of foods that presumably benefit the gut microbiome on a regular basis? Is there any evidence to suggest that?
If we change the narrative slightly, I would argue that fermented foods generally have beneficial impacts because they have both a antigenic stimulating component, which can improve immune system function, but they also have microbial products from their fermentation that are both absorbed inside the body and can actually feed other bacteria inside the body. So I would say the consumption of a diverse plant diet, so you should try and consume at least 30 species of plant every week, is a really good rule of thumb. And also the consumption of fermented foods can be a really nice way of starting to improve microbial health and total physiological and immune health. maybe hormonal.
Very helpful. And actually, a related question for Dr. Gabel. Does TRE work because it results in calorie reduction alone, or does what you eat matter?
Well, I am a dietitian, so I do believe what you eat matters. That's my new goal. That the number of vegetables might be my new goal, or plant species. I'm always trying to make sure I get my 5 and 5, but I think the reason it works and is accessible currently is because it is calorie reducing. Now, is that I think this is a good step, right? It's a good first step to be at a calorie reduction. Would I love for people to change the quality of their diet? Yes, but there are a lot of other factors that influence people's ability to do that. So to me, this is a diet that's accessible and that anyone can do regardless of their access to food, their financial ability to buy different kinds of food. So again, I think it's a good first step, but I would obviously love people to eat more plants.
Thank you. Professor Gilbert, are there ethnic differences in the gut microbiome?
Absolutely. A lot of them are driven by changes in lifestyle patterns rather than any genetic association to, say, a change in melatonin level in the skin or historical differences in other factors. It's immune history of those individuals and lifestyle of the individuals that appears to be the biggest driver. But we do currently have a population of 2,500 people of African origin, from 5 global countries, including 3 in Africa, that is allowing us to have insights into cardiometabolic health. And now, because our population is aging into menopause, menopausal health in these individuals across what we refer to as the epidemiological transition from countries like Ghana, which live a very traditional lifestyle rich in fiber and polyphenols and those fermented foods. You'll be happy to hear, Kelsey, and all the way through to America, and especially in Chicago, where a lot of our participants have a less desirable diet, if you will. And so there's a transition there which we're investigating to determine its impact upon these factors.
Great, thanks. So final 2 questions. Dr. Gabel, how can we keep up good spirits while fasting? People on this TRE tend to get cranky. Is there a supplement for this?
I think people on any calorie restriction are cranky. I would say trying to find the window that works best in your life. And if you need to shift. So for a lot of people, having that eating window in the afternoon to evening allows them to have more of a social life, eat dinner with family and friends, things like that. But maybe on the weekends, you want to shift it to earlier in the morning. I had a lot of participants that on Sunday mornings wanted to eat brunch with their family earlier, so they would shift that window and have an earlier dinner. So really, I mean, thinking about giving yourself some wiggle room around when that window is can benefit kind of some of that mood.
Thank you. And then a question for Professor Gilbert. I understand that there is data to show that intermittent fasting improves the gut microbiome. Is this true? And if so, does the window make a big difference? Thanks.
So actually, there is some interesting data to suggest it increases the diversity of the microbial community, and we're not entirely sure we understand why. We do know from animal studies, mice and rats studies, that intermittent fasting appears to increase short-chain fatty acid production, especially butyrate levels in some of these animals, which can alter both insulin and incretin secretion sensitivity inside the pancreas. So there is some relationship there to health characteristics, but we really don't understand it as effectively as we should. We do know that at the beginning of the day, after your night fast, you have a significantly reduced abundance or biomass of the microbiome in your gut. And then whatever you eat first during the day appears to promote the growth of organisms in that reduced biomass that are either beneficial or not beneficial. So the first meal of the day appears to be the most important meal for stimulating the correct kind of microbial activity. So it would suggest a high fiber, potentially plant-based meal at the beginning of the day, rather than something very sugary or high in saturated fat.
Wonderful, thank you. We had more questions than we had time to answer, so a lot of interest in these topics. I'd like to thank you both very much for these informative presentations and for your responses to the audience's questions. Many thanks to the audience for attending today, and of course this will be as a webinar on the IMS website for those of you who want to share it with other registrants. So thank you again, Professor Gilbert. Thank you, Dr. Gabel. And thanks to the International Menopause Society and Bessons. Okay, and with that, I wish you all a very good day.